start in just a little bit. A few more people will be logging on, I'm sure. I'm going to mute for a second.
Okay.
All right, I think we can get started. We might have a few more people trickle in, but we'll go ahead and get started. We welcome everyone to Geriatric Medicine Grand Rounds. Happy New Year. It feels kind of weird saying that because we all know that our current times are quite unusual and challenging, but Happy New Year nonetheless, and I hope you all had a little bit of time to refresh before we started the new year. But we have a wonderful speaker today. Dr. Jonathan An is a assistant professor in the Department of Oral Health Sciences. He's the assistant director of the graduate program in oral health sciences. And his research work is why I invited him here today. He really works at the intersection, the relationship between biological mechanisms of aging and some common oral health conditions, and just really fascinating work. He was named a Magnuson Scholar to do this work. And received a graduate training award previously from the National Institute of Dental and Craniofacial Research. He's done quite a bit of work and it's really fascinating. And I'm really excited that you've come here to share with us.
Great, thank you, Kate. Yeah, I appreciate the invitation. So my name is Jonathan An. I like to describe myself as a dentist who is also a scientist that studies the biology of aging in the context of oral health. Before I start today, I just want to say I have nothing to disclose. So this is just my disclosure statement. And so just kind of the topics I want to describe today and just kind of the agenda for today. First, I'm going to describe a little bit about the increase of age as well as the aging nation currently that's happening, as well as how oral health and disease plays a role in this older population. And then I will discuss a little bit about the geroscience hypothesis, as well as what this is and its impact on oral health and disease. The first kind of 3 points are going to really heavily rely on some of the basic science work that my laboratory has been doing. But ultimately, what we're going to go into is talk a little bit about some of the translational geroscience work, and especially work that we are completing now, as well as some projects that we're about to embark on. So I don't really have to kind of belabor this points to this audience here, but the population is actually aging, and for the first time in about 2035, Americans over the age of 65 is going to outnumber the children who are under 18. And obviously, with this demographic shift, there's going to be multiple areas where it's going to be impactful, not only socially, but also, for example, economically. So this is actually data, and this could have changed because of these recent events that happened, but at least in 2020, what they've shown here is that the total healthcare expenditures, which are in billions, and you're looking at here at the bottom line for older adults over the age of 65, has more than doubled in the 20 years. So in addition to the economic impact, as well as social impact, age is also the greatest risk factor for many known diseases, as well as a leading cause of death. And so some of these diseases that are heavily age associated include Alzheimer's disease, cancer, diabetes, as well as heart disease. And so how do these kind of systemic as well as local age-related diseases, and how does dentistry or oral health fit into that? And actually, if we take a look at some of the commonly prescribed drugs that are reported in dental offices, if you just do a quick scan of all these drugs, majority, if not all these medications are taken because of some form of chronic condition that affects older adults. So, lisinopril for high blood pressure, hypertension, Simvasan for cholesterol, as well as type 2 diabetes. And so the dental office, we actually see very routinely many older adults, and some of you might have been going to the same dentist for over the years. But in addition to this impact, the other impact is also economically. So here you're looking at the same kind of graph, but now we're just looking at dental healthcare expenditures overall. And now you're looking at the top line here, whereas more than doubled in the past 20 years for the older adult population. And so what are some diseases as well as declines that actually affect the older population? And prior to this year, I kind of grouped various different articles into kind of multiple slides and kind of describe kind of these age-associated oral diseases in health. But most recently, the NIH as well as NIDCR kind of helped out, and they actually updated it for the first time. I think their first report was back in 2000. So it's a 20-year update where they really explore the oral health in America. And I think it's about an 800-page document. And the neat thing what they did was they actually divided it up by age. And so there's about 70 pages of that, or 60 to 70 pages related to older adults. And they just highlight all the different diseases and unanswered questions that we still need to do. And so if we go through this list from this report, we find that a variety of common conditions that really hasn't changed in the past 20 years that affect older adults. So dental cavities, caries, periodontal disease, tooth loss, oral cancer, oropharyngeal cancer, pain or TMD, as well as low saliva or salivary gland dysfunction. And I want to take a few moments and kind of describing a few of these. And then for one of these, I'll go into a little more detail later on. So first is oropharyngeal cancer. And so this is cancer within the oral pharynx, which is the middle part of the throat here. It involves the back of the throat, base of the tongue, palatine tonsils, as well as a soft palate. And because of this, we're trained as dentists to not only just look at the teeth or inside the oral cavity, but we complete a full head and neck exam during every visit, but also an extraoral as well as an intraoral exam for oral cancer, such that we don't miss something like this. And obviously this is a really severe case of oropharyngeal cancer. And once a patient gets diagnosed, actually the percentage of deaths actually increases by age. And so the median age of death is about 68 years old. And again, this is from the same report. Again, NIDCR and NIH really helped me out on this. And so again, the percentage of deaths by age group does increase. The second type of common oral condition is periodontal disease. And this is something that I'll kind of describe more later on. But periodontal disease is an oral inflammatory disease that's marked by gum inflammation, loss of supporting tooth structures such as the bone, causes bleeding gums, as well as painful on chewing. This is a little bit different from another common term you may hear called gingivitis. And gingivitis is just inflammation of the gum tissue. And this is reversible, but as you can see, this is not. If we take a look at just our nation, overall, about 65 to 70% of older adults have some form of periodontal disease. And so this is just a graph in the spectrum of green where, for example, in Washington, you're looking at about 64 to 65% of older adults having some form of periodontal disease versus somewhere in California or even in Texas where it could go up to 70%. For the next one, I want to kind of combine both kind of the aging biology as well as kind of oral health a little bit more. And so I want to describe cavities and low saliva. And you might be wondering, okay, how are those two connected? I thought cavities are related to flossing, and plaque buildup around there. But I'll kind of try to show you the direct correlation between the two. So when we see in the clinic, sometimes older adults will come in not knowing they have low saliva or dry mouth, or they will explain that, Dr. An, I've been always having to drink more water during my meals. I get this sticky substance in my mouth. And so usually that's a sign of dry mouth or low saliva. And what this actually causes is what's called plaque accumulation. And so plaque is that kind of that sticky, kind of yellow or colorless kind of biofilm plus, some of the food debris that you ate that gets stuck on there. And the reason is that saliva not only does it act as a lubricant and it has some antimicrobial compounds in there, but it actually flushes a lot of those plaques around so that it can be either swallowed or removed from the teeth. And so when this plaque actually accumulates, what happens is a cavity that's commonly found in older adults called root cavities. So these are cavities that happen right around the gum line or by the roots. And so as clinicians, what we do is when we see the cavity, remove the cavity, and we do a filling or a restorative filling. And our field has done incredible work in developing proper restorative materials, bonding agents, as well as this whole process so the filling stays. However, we still haven't really solved the real issue that's why this patient had to get the filling done. It could have been the plaque accumulation, but really what it ultimately was is low saliva or dry mouth. So even when this older adult or this patient gets a filling done, we still haven't solved this low saliva. It still causes plaque accumulation, and the patients end up getting what's called recurrent cavities or cavities already at the site of where the restoration is. So it becomes this kind of cyclical cycle for our patients. And so if aging physiology plays a major role in not only, if not all, chronic diseases, including oral disease, then if we could address aging directly, it may prevent the onset as well as mitigation of severity of multiple chronic diseases at the same time. And this is actually the essence of the Geroscience hypothesis, which is that rather than looking at all these diseases at an individual level, if we can understand the process of aging and target this process, we may be able to reduce the impact of all of these diseases simultaneously. Because ultimately, the optimal functionality for any single system actually requires overall organismal health. And so are there ways that we could actually intervene and kind of target this process? And there's a few out there, but one particular intervention I want to discuss today a little bit about is a drug called rapamycin. Rapamycin, at least in model organisms, is the most robust as well as most studied and reproducible geroscience intervention that's not only seen to increase lifespan, but also delay age-related phenotypes. And some of you may be prescribing rapamycin or mTOR analogs in your clinics for other various reasons. But rapamycin was first isolated from soil samples on Easter Island. Its name is actually derived from the island it was found on, called Rapa Nui. And it was initially developed as an antifungal agent, but has been shown to have a potent immunomodulatory impact. And genetic studies in yeast first identified this target of rapamycin, and that target is a gene called mTOR, which actually stands for mechanistic target of rapamycin, so mTOR. And mTOR is a nutrient and growth factor responsive kinase, and it's structurally as well as functionally conserved from yeast all the way into mammals. And so there's a variety in multiple studies showing the impact of rapamycin on model organisms, but one particular study I want to highlight is actually a study done by the NIH at the National Institute on Aging via their interventions testing program. And so the ITP is this collection or multi-institutional program that tests interventions that are proposed by the scientific community that may impact lifespan or even health. And each of these centers actually performs their own lifespan analysis on a genetically heterogeneous UMHED3 strain. So what a lifespan analysis pretty much is the intervention is given to the animals at a certain age, and the animals are actually followed throughout their entire life until they pass away. And that's the essence of what a lifespan analysis is. And so if we actually take a look at the data for rapamycin, and so this is a survival curve, and where you're looking at survival at 100% to 0, both in males and females, with increasing doses of rapamycin, so from 4.7 all the way to 42 parts per million, compared to control, both males and females show an extension of life as a function with increasing dosages of rapamycin. I want to highlight that this rapamycin is actually given within the food. So it's mixed in with the food, and the animals are on the food for the duration of their life. But in addition to lifespan, rapamycin has been shown to delay multiple age-related phenotypes, such as cancers, declining muscle function, cognitive decline, as well as improving immune response. And so the question that we had in our lab was, is there an age-related oral disease that's kind of impactful as well as correlated to other age-associated diseases that we could potentially target to improve its outcome? And that disease we initially started off looking at was the periodontal disease, which is, like I mentioned, a chronic oral inflammatory disease. And this is just the same data, just showing about 65 to 70% of adults, older adults, having some form of periodontal disease. And by definition, this disease is inflammation that happens around the supporting tooth structure that leads to bone loss with a variable microbial pattern. And many times periodontal disease is diagnosed in their own dental clinics by two ways. One is clinical examination, which I'll describe here in a little bit, but also dental x-rays or radiographs. So, and the bone loss is actually the irreversible part that happens during periodontal disease, where at normal bone levels and with the inflammation and periodontal disease, you have this loss of bone. And so, as with many kind of basic science research inquiries, we actually wanted to first come up with a model to evaluate the disease and its process during age. Ultimately, our goal is to really develop feasible as well as practical and safe interventions to delay disease and conditions in humans. But we need to have suitable models to that. So what we first started off was looking at the rodent model, which is commonly used in oral biology as well as in aging research. And, a little bit different from past research on periodontal disease is that, prior studies actually have utilized artificial disease models in younger rodents, which actually try to mimic this age-related phenotype. But a limitation in these studies is that we actually lose the contribution of the truly aged local as well as systemic environment. Thus, in our studies, we don't induce any artificial disease, and we just wanted to evaluate what happens during normative aging. And so if we take a look at, for example, we could take a dental x-ray on mice using a micro CT imaging. And if we compare, for example, a young mice, so this is a mice that is about 6 months old, and this is the top of the teeth of the mice. So mice only have 3 molars on the top as well as on the bottom. And you compare that to an old mouse. So an old mouse now is about 22 months old, so about 2 years old. You could just visually see, so this is the bone, the periodontal bone on the top teeth and the bottom, and this is some of the root structure showing through as well as the tooth. Just age alone leads to this kind of periodontal bone loss around the teeth, both on the top as well as on the bottom here. And if we isolate the gum tissue and the bone within these animals and run an inflammatory panel, we could see, so on the top, you're looking at the gum tissue, and on the bottom, you're looking at the periodontal bone, and our baseline is at 1 for both, and these are the younger animals at baseline 1. We can see that with just age alone, there's a variety of inflammatory cytokines and chemokines that are increased in both the gum tissue as well as the bone. So as we uncovered that age actually causes this common oral disease, we wanted to actually evaluate now whether or not targeting the biological aging process could delay periodontal disease. And because this is the first time the study was being done, we wanted to make sure it was robust. And so what we did was we did this cross-country collaboration with the Jackson Laboratory, where both sites actually raised the animals, and we had a young and adult cohort just as more of our baseline control. And then we also had an older cohort that were either given, again, this is within the food, either a controlled diet or diet with rapamycin for 8 weeks, and then the tissues were harvested. The other aspect that we did was we included in the Jackson Laboratory cohorts an initial baseline scan of the micro CT. So we're looking at the baseline levels of the bone. And then after treatment, we also took a final micro CT just to look at if there's any longitudinal changes within the same animal. And so how we actually analyze our data was using the very definition that we use in humans to look at periodontal disease. So we looked at the gum tissue, we looked at the bone, as well as look at the microbiome. And I want to kind of explain a little bit about each one now. So first, we wanted to look at the gum tissue. So gum tissues or gingival epithelium is actually one of the first line of defense against biofilms, food, and it's in constant immunosurveillance with the commensal bacteria that are native to your oral cavity, as well as other inflammatory responses that are happening within the oral cavity. Now, inflammation in the gingival epithelium can actually be visualized pretty clearly in humans or in a clinical evaluation, but it becomes a little bit challenging in a mice model. And so instead, what we ended up looking at was actually molecular hallmarks of inflammation through the NF-kappa beta pathway. I'm not gonna really go into detail of the NF-kappa beta pathway, but essentially this is a key role in regulating immune response. Basically what happens is it's in a heterodimer mix. And so once this IKB alpha, phosphorylated IKB alpha gets degraded, these two proteins then bind to their specific sites, leading to an inflammatory response, increasing mRNA inflammatory gene as well as protein expression. And so when we look at the gingival tissue, so on the left, I'm showing you a protein blot looking at NF-kappa beta as well as IKB alpha. And on the right, I'm showing you the inflammatory panels like I shown you before. So if you look at just P65 and IKB alpha, we see that just with age alone, in the old animals in their gingival tissue, we see an increase in P65 with the degradation of IKB alpha, and that the old animals treated with rapamycin actually showed an attenuation of that P65. And in addition to the increased inflammation in just the old animals here shown in pink, the blue here now show the animals that were treated with rapamycin showing attenuation of this inflammatory cytokine. Next, we looked at periodontal bone. Again, visually we could look at periodontal bone, but it's a very intricate process, bone homeostasis, in that it's a fine balance between bone-forming osteoblasts as well as bone-resorbing osteoclasts. And with more rankle presence in there and more rank binding in that, and actually activates more osteoclasts. And in fact, if you look at periodontal disease, there is an increase in rankle activity that is happening compared to OPG, which actually protects the bone from resorbing by binding to this rankle. So if we actually look in our old animals, we actually find that same thing, where in the old animals, we see an increase in rankle activity, and that rapamycin actually attenuates that at the protein level. And similarly to our gum tissue, what we find is that compared to the old animals treated with rapamycin had a significant attenuation of a variety of inflammatory markers. But the one thing nice about bone or the micro CT imaging is actually we could visually see what's happening. So this is just, again, the top teeth on your left here between an adult animal and an old animal, where you could see in the old animal, there's loss of bone around the roots here. And then this is the lower teeth or the mandible here. And visually, what we could see is that in the old animals given rapamycin, some of this little area here are now filled in with some of this hard deposits, as well as in the mandible bone. Now, if this was in humans, or even if we could do these in primates, we could actually do a clinical assessment of periodontal disease. And many of you might be familiar with this. And we could actually use a periodontal probe, where maybe your hygienist or dentist, looked at your gum pockets and read numbers like 2, 3s, and 4s. And essentially what we're looking at is this pocketing, where if there's bone loss as well as inflammation, there's increasing pockets. So simply put, the lower numbers you hear is actually better. But we can't, again, do these in rodent animals. And so what we ended up doing was going into 3-dimensional space. And what we did was we figured out there are common landmarks, anatomical landmarks that we could identify within the mouse teeth. So this is the cemento enamel junction, which also is present in humans, as well as this alveolar bone crest. And what we could do in this 3-dimensional space is actually landmark them at various different points along, and actually measure them just like we do with periodontal probing. And this is actually done by multiple people, like 3 to 5 people, and then the data is pulled together. So a clinical evaluation can actually become a quantification. And so what here you're looking at is the periodontal bone loss on the left axis here. And so the more distance there's actually more bone loss. And so on the top teeth, the maxilla, as well as the mandible, with increasing age, we see that there's increased periodontal bone loss. And then rapamycin treated old animals showing attenuation. To really kind of highlight this point is that we could look at the longitudinal imaging. So on the left, you're looking at a before scan of an old animal given rapamycin. And so if you look at this particular imaging of the mice, you can see that between the roots, there's bone loss, where you could clearly see this cul-de-sac here. On the top teeth, you could see where in between the teeth, there's bone loss, as well as you can kind of see a concavity because of that bone loss, as well as loss in here. But in the same animal, after treatment with rapamycin, you can see now this area is being now filled in with this hard deposited bone, as well as in between the teeth here. And in fact, also on the bottom teeth, on this top panel, you can see now this area is now covered with this hard substance here. So it almost looks like there's this kind of regenerative effect of this periodontal bone in these animals. So in addition to the molecular and clinical evaluation, one of the last components in periodontal disease is the oral microbiome. And I'm not gonna bore you to details with the sequencing data, but one data I do wanna show that really showed that rapamycin actually caused this reverted shift is this data on beta diversity. So beta diversity is what we're looking at, is how different the microbial composition is from one environment to another. And so if you look at the oral cavity of a younger animal here in the turquoise, we can see there's a shift between the younger animal versus the old animals here in the red. However, if we look at the rapamycin treated old animals, which is here in the blue, or it could be a little bit different on your screens, they seem to overlap with this younger animals, which indicates that there is this reverted shift of the aged oral microbiome by rapamycin to be more similar to the younger oral microbiome. So if we go back to the very definition of periodontal disease and look at these 3 parameters, we find that rapamycin actually targets all 3 simultaneously. Some continuing work that we're doing now and preparing for is that, what happens after the 8-week treatment? Is this just a one-time and it just stops, or does the results actually persist after? And so we kind of followed up with this study, and we're finishing up this now, is that what we had a cohort of animals first given a controlled diet for 8 weeks, and then we gave them rapamycin just to kind of mimic and as more of a control cohort for our initial rapamycin study. And then we had a second cohort given the rapamycin initially for 8 weeks, and then they were off the rapamycin with just to be on the controlled diet. And I'm just going to show you 2 data that I've shown before. First, the wrinkle data at the protein level. And what you see is between the young and the old, again, there's an increase in wrinkle. Rapamycin for 8 weeks decreased the wrinkle activity. But in both of these cohorts, we see, again, there is this attenuation of wrinkle activity, even the ones that were given rapamycin off the drug for 8 weeks. So it seems like there's a persistent inhibition of wrinkle in these older animals. In addition to that, when we looked at IKB alpha, again, compared to the old and rapamycin, we see that there's this attenuation of phosphorylated IKB alpha. And again, the results continue to persist. But really, to highlight that is really the micro-CT imaging. And so you can look here on the baseline data before treatment, you can see the bone loss around this particular root structure here, and just so that you can see the concavity as well as the furcation. And after 8 weeks, just like we've seen before, we see some of these areas now being filled in with bone, as well as around the root structure here. And then if we take a final CT imaging after the treatment, we can see that while there is still some bone loss that occurs even after, the bone depositions are still present. So it seems like it's persisting even after the rapamycin has stopped. And this is just the quantitative data. And so obviously we're finishing up the study, and we're actually seeing the same kind of responses in the inflammation markers. And so hopefully we get that published here very soon. I want to shift gears now from the basic sciences to now really kind of translational work as well as models for geroscience. So obviously what we've shown so far is that in older mice, we can actually inhibit the mTOR by rapamycin and actually reverse periodontal disease. And ultimately, what we want to do is help in humans out, but it becomes a little bit challenging in terms of, how we get there. And so one thing we decided to do, and obviously many of you may encounter, is this jump is very difficult. And so we've been denied multiple times when our grants. And so we try to come up with an intermediary. And so we had an interesting collaboration with some groups looking at actually non-human primate models. And I'm going to show a little data from this, not too much because we're still kind of finishing up this work. But what we did was we looked at younger females and older female animals. And we started off with females is because the data I've shown you before is actually all in female mice, at least for now. And we actually gave the animals a 1 week dose, a very low dose of rapamycin for 8 weeks. And then we assess kind of the same parameters. But what we could actually do is actually do dental exams like we do in the clinic. So we could take dental x-rays, and we had that set up we had that set up in the facility to look at periodontal bone loss, as well as we could actually do clinical assessment of periodontal measures such as probing depth, or even what's this measure is bleeding on probing. And so what this measure looks at is the probing, periodontal probe is actually left in there and then removed. And then we kind of look at whether or not these sites actually bleed, which is indicating of inflammation. And so this is just some radiographic data. So on the top, I'm showing you that the mice data. So again, in the younger animals, here's the difference between the two landmarks. And then the older animals, you can see the periodontal bone loss. And this is actually true both in humans as well as in aged animals, where you can see in the younger animal, the younger primates, you can see there's less bone loss. And just with age alone, in about a 19-year span, there is this periodontal bone loss occurring. And if we look at just periodontal probing measures, so on the left, we're looking at periodontal index, compared to the baseline where the younger animals are, there's a significant increase in bleeding on probing in the aged animals. And in fact, during the whole treatment process, we actually find that the bleeding on probing index actually decreases. So it seems like there's less inflammation going on. And actually, if we pull kind of all the age data together, we find that during the rapamycin treatment course, we find this to be significantly decreased. And we're actually running some of the gingival fluid that we collected, as well as some of the saliva. And so hopefully we have some more data within that. And so the other aspect I want to really highlight too is obviously we want to go towards helping out humans. And over the years, there's been multiple studies or primary literature just showing that there's a high translational potential for using rapamycin as well as other mTOR inhibitors to improve health in older adults And so, for example, this was a trial that's completed most recently where they gave healthy older adults a dose of 1 mg of rapamycin daily compared to a placebo, just to look at feasibility and safety. And I think overall, their study showed that they ran standard panels for metabolism, clinical laboratory panels like CBC and HbA1c, as well as glucose tolerance tests. And they actually reported there's no other adverse events. And actually, both in the placebo and the RAPA, they had very similar. I think one subject had adverse events. It was the same adverse event. And they saw some changes in erythrocyte parameters, but it didn't really manifest into any clinical manifestations. But in addition, in terms of immune response, this is done by Joan Manning's group, where they actually gave rapamycin, or not rapamycin, an mTOR inhibitor, everolimus, to patients right before giving a influenza vaccine. And what they found was that the older adults that were given everolimus prior to the vaccine actually showed a better immune response to that vaccine than the older adults that were not given everolimus. And then most recently, this group showed that if you apply topically rapamycin, it reduces markers of senescence or human aging. And so really what we ended up wanting to do was, try to find out, what is the impact of rapamycin on periodontitis in older adults? Because ultimately our goal is translating some of our basic science findings to humans. And after getting, multiple denials, we were at the end of last year. So while I was preparing for this presentation, we were told that we got one of the Longevity Impetus Grants Awards at their max funding level, and to actually proceed with this now clinical trial here. And so we're working with the ITHS as well as the School of Dentistry to do this. And then I was telling Diane early on that we're hoping to find some interesting groups to work with within the UW Medicine. I've gotten a lot of guidance from Dr. Angela Hansen, as well as Dr. Deb Huang. And so we're hoping to kind of start that off this year, and we're busy going through all the investigator-initiated clinical trial process now. So in conclusion, what I want to describe is, age also impacts the oral cavity, and studies understanding or aging hallmarks to extend the oral health span is critical. What we've shown, at least in the basic sciences, is that in aged mice treated with short-term rapamycin reverse 3 major clinical hallmarks of periodontal disease. And ultimately, what I think is really neat is that, this whole project supports the Geroscience hypothesis, showing that interventions that target the biological aging process can simultaneously delay multiple age-related disease and functional decline. I just want to acknowledge my collaborators, my students, as well as my funding sources down here. And then with that, I was told to put the QR code up there. I'll go ahead and take any questions that you may have. Thank you.
Can I jump in, Kate?
Yes, you're the boss. Yeah.
Hey, John.
Hi, Wayne. Hello.
That was terrific. Thanks so much for doing that. My job as division head is to ask the stupid questions that everybody else is too embarrassed to ask. So here they come.
No.
There are 3 related questions. One is, well, first of all, these experimental mice obviously were not flossing, so we're clearly disappointed in them for that. But our patients often ask us, is it good to floss? How often? Can you floss too much? Obviously, you can floss too little. And actually, there was a study about a year or 2 ago that among your colleagues that said something about flossing not doing any good. So that's question 1. And then the other 2 are related, and that is we see lots of marketing about rinses for older people and artificial saliva. Any recommendations about that? And particularly about xylitol that tends to increase the price of those items, good, bad, or indifferent? So those are my stupid questions for you.
No, they're not stupid questions, and they're very challenging even for me, both not only as a dentist but also a scientist, because obviously we want to do what's really evidence-based, right, at the end of the day, and most importantly. To your first question about flossing, I do know what the paper you're talking about, and so I could tell you that it's not going to stop me from flossing. I know that in the article they described a lot of the trials that were showing that flossing was not effective, or that it did not have an improvement and benefit. For those who are doing degrees, the dentist side of me, we follow kind of the ADA recommendation, which is the American Dental Association recommendation. But the science side of me, when I see that is very good research. The practicality of that, I can't really comment on. But like I said, it's not going to stop me from flossing, and I don't know if that's a good way to do it. In the clinic, I do see patients quite often that don't do any of the recommended oral hygiene, and they have really good oral hygiene, right? So it's one of those clinical kind of questions that I don't know how to answer. But so I don't know, there's no way to directly answer that. But if a patient does come in, that is my recommendation, is at least continue flossing, because it's been shown to, at least in terms of plaque removal, that's effective. Whether or not that leads to other things, I don't know.
Well, let me jump in, John, and say that that study was among the general population. Right. You've elegantly illustrated how prevalent periodontal disease is in older people.
Right.
So yeah, it's conceivable that flossing could be harmful in people who have fairly advanced periodontal disease, or maybe it's really good for you. I'm wondering which.
Yeah, the tricky part with periodontal disease is that with the probing depths, with the pocketing that these patients have, a lot of times the flossing can't really go down in there. And so a lot of times that's why for patients, we actually recommend coming in every 3 to 4 months for the maintenance cycle to kind of remove the biofilm within those pockets. And so in a way, I don't know. I mean, I know some people that do overfloss, and we see a lot of gingival trauma that happens. And I think it's just at that point, some patients are really concerned about the oral hygiene. But yeah, I don't know how to really appropriately address that question. In terms of rinses, oh, sorry, go ahead. I'm sorry. In terms of rinses, like you asked, or even xylitol. So xylitol does have some anti-caries benefit, as well as kind of inducing, like you said, within the artificial saliva. These are one of these things where, companies will approach the clinics for marketing purposes, and they'll give us samples to do. I've seen a wider range of efficacies in patients. Some patients don't like the artificial saliva because it is artificial. It's very sticky. And, I mean, we do recommend it. And I don't know if you ever tried it, but I try to use the product. I try to recommend to my patients, and it's, maybe it's because I have lots of saliva too, but it's not again, I have no conflicts of interest with this, but it's not a really pleasant kind of experience. So usually for those kind of patients that work, that's great. If not, we recommend kind of like, like mints, or there are these lozenges that patients can do. And then, yeah, it does increase the price for a little bit. A lot of times you could get these like xylitol mints over the counter, at really famous kind of online internet shopping place you could get it at. But when new patients come in, we like to first assess, what causes the low saliva, for example, whether it is just age related, whether it's medication related, or whether it's radiation initiated. And then for the most times, unfortunately, there isn't really a good substitute, and patients just kind of live with that, which is the unfortunate part. I want to add a little bit on there that the other aspect that we're working on is we actually find in old mice that low saliva actually significantly decreases when we actually look at saliva flow in the old animals right now, and taking a look at, okay. Yeah, so again, I don't know if I answered all three questions, but yeah. Let me see.
Great, thank you.
Yeah. We have some questions in the chat, and then I see that Dr. Grunwald has his hand up too. So maybe we'll get to a couple of the questions in the chat first, if that's okay.
Sure.
Dr. Stephen Playmate was wondering if there were one or more specific species in the microbiome that were dominant in relation to bone loss.
Yeah, so the depth of the sequencing, I mean, we try to get pretty far in terms of, are there specific genuses or phyla that actually matter? So what we're finding is actually it's actually impacting some of the bacteroides, which is one of the common microbiome family that actually induce in periodontal disease. Specific species beyond that, we don't quite know yet. And that's just, more questions that we have to ask, but at least the bacteroides is something that we're finding both in mice as well as in human studies has shown that. So, yeah.
Other questions from our research colleagues. Dr. Lindsay Anderson was wondering if you've seen any effect on muscle mass or physical function, since mTOR is part of the primary protein synthesis pathway in skeletal muscle.
Yeah, so I have not seen it because obviously I'm more focused in the mouth, but I know there's a lot of researchers that actually look at that. I think from literature, I think it does, rapamycin decrease the number of myocytes, I believe, but other than that, I don't really know in terms of how that affects muscle mass. What I do know, and I don't know if this is correlated, is that the animals that do get treated with rapamycin do a little, at least injection, they do lose some weight. So maybe it does have some impact on mass, but beyond that, I don't know. So sorry, I don't, yeah.
Oh, that's great. Thanks for answering that question. Oh, I'm being asked if you could put the QR code back up.
Oh, sorry. Yeah, sorry.
Do
I will
that.
go ahead and, yeah, let me put that back up.
While we're doing that, Dr. Elizabeth Ray is asking if there's any evidence for beneficial effects of rapamycin on other diseases related to bone loss, or
Yeah.
aging. Such as, or an impact on frailty.
Trying to think if in terms of bone loss, osteoporosis, no, I'm not quite familiar. No, I'm sorry. I don't know. I'm pretty sure. Actually, there is studies that show that rapamycin impacts trap activity. So trap is osteoclast activity within the bone structure. So I'm pretty sure there's got to be literature out there showing its impact on there. Yeah, I'm trying to, I know of a paper, but I don't want to misquote the findings of that paper, but they did look at various osteoclast activity, and I think vertebral columns, and they actually find that, yeah, sorry, I don't want to misquote the article. I'm sorry about that. Yeah, but yeah.
Thank you. And then Dr. Gridwell, you've had your hand up for a while. It's probably getting tired that hand of yours. Go ahead.
Yeah, thank you. This is just fantastic information and line of research. I'm wondering if there are data on everolimus or tacrolimus, which you mentioned at the end of your talk, sort of thinking about almost a pseudo experiment in people who receive agents like this. Do they have any beneficial effects on periodontal disease?
Yeah, no, that's a great question. So initially, when we thought about kind of looking in older adults or the population, we wanted to kind of do that where are there patients taking these medications and doing that. Many times, as you may be familiar too, is that these patients are not just on these mTOR inhibitors. You know, they've had, for example, an organ transplant, and they have these other comorbidities, and they're on multiple medications. So really assessing whether or not that's an impact of mTOR inhibitors, what was kind of a challenge that we encountered. However, there is a group, and it's actually Matt's group now, where they're actually looking at healthy older adults that were prescribed rapamycin for some reason that didn't go through an organ transplant or may have, and actually really trying to assess the safety profile within that. So initially we were going to go through the UW system and seeing if we could reach out to these patients and kind of do more of a survey-based analysis. But because of the confounding factors, we didn't do that. But I think Matt's group is going to go ahead and do that in terms of survey-based around the country now. So hopefully we'll have an answer. But yeah, that's a great point, David. And that's something that we'll have to look into. Especially, I mean, we want to look at potentially healthy older adults who may be taking mTOR inhibitors or rapamycin. So yeah, that's a great question.
Thank you.
Yep.
Oh, I have a question too. Yeah, I'm sorry if I missed it, but I assume you're studying other interventions, right? Yeah, besides, I mean, I'm sure there's so many different ones, and our patients are dabbling in a lot of them. And some of them overlap with mTOR pathways, of course, but others don't. Just wondering what else you're cooking at the same time, because I know you all are. So.
Yeah, yeah. So yeah, there's a couple of them, Mary. First thing we're actually looking at is along the PI3K of Kappa beta pathway. There are kind of specific inhibitors as well as specific agonists that are available that we could test. That are one of our collaborators actually using some knockout models for specific diseases have shown some pretty beneficial effect in those kind of compounds. And so we're actually testing them out in oral cavities. So along kind of the inflammation pathway. The other thing we're actually looking at, Mary, you're probably familiar with, is also the senescence pathway. So various compounds that target cellular senescence. So for those of you who don't know, cellular senescence is an irreversible cell state. And the idea is that the cell actually doesn't die out, it actually kind of continues there, and it produces a lot of inflammation and a lot of side effects. And so what groups have shown is that if you actually target the cellular senescence pathway in aged animals and mice, you could actually extend their lifespan. And what we actually find is that in that genetic model that we actually found, and it's unpublished, but that the periodontal bone loss is actually attenuated. So I think there is some overlap between the mTOR pathway as well as senescence. And obviously there are pharmaceutical compounds now that actually target the senescence pathway that we're looking at. So, nasonab quercetin is one, DNQ is one. There's some other ones like ABT that groups are looking at. And so, those two. And then the other thing we're kind of doing right now is reevaluating some compounds that have been proposed actually in dentistry for older adults that haven't shown to be effective. And I think one reason is because of using the younger animal model rather than the older animal model. So we're also kind of reevaluating some of these kind of older interventions or older kind of genetic pathways that really didn't show an impact in the younger animals in the older animals. So there's a lot we're trying to do. It's always a matter of time and people to kind of help you out. But yeah, hopefully that answers your question, May.
I have one more question. Do we know, you may not, maybe someone knows, does anyone know if the big Metformin trial is going to have oral health endpoints? We're talking about TAME, of course.
Yeah.
Aging with Metformin.
Yeah, I don't think they do.
I know. I didn't hear of any either.
Yeah, when I heard about the trial, I wish they had, worked with the NIDCR on that.
Yeah.
Because they had I think it would have been a very interesting trial to do, just kind of look at everything. Yeah, because I think the oral health is such an invasive kind of non-invasive, really quick way to assess kind of the oral cavity as well as this phenotype. And so, we were actually I don't know if you're familiar with Matt Caberline's dog aging project. So we were actually thinking of maybe looking at oral health in dogs because
oh my gosh, yeah, save us a lot of money.
Yeah, number one thing that happens in dogs is periodontal disease, right? So that's something that we're looking at, but there's some nuances where the dogs have to actually go under anesthesia.
Yeah.
But yeah, that's the other thing we're here for, May, is that if there's any questions in terms of the oral cavity, oral health, we can dissect that and we can look, and we have all the team to do that.
Well, can't you just contact the TAME investigators and ask them? Because my understanding is that they have so many endpoints, and it's a matter of having the expertise to track.
Yeah, that's something that we will probably end up doing. But yeah, that's a great suggestion. So I don't know if I think most people are familiar with the TAME trial, but they're looking at metformin, where they found data showing that individuals on metformin actually have a longer life, pretty much. And so now they're going back and really doing a complete trial on this. So that's just what the TAME trial is.
Great. Well, we have a few minutes left if anyone has other questions, although we've asked you lots and lots of questions. Probably
this is great. Yeah, the one thing I really appreciated being here on campus is, just the broad and just the wide scope of, fields that you could encounter. We're sometimes stuck in our own little niche that, we don't expand out. And this is great for me as well. Yeah. So
you mentioned, looking for opportunities to collaborate with our division on your second to last slide. And I was just wondering if you wanted to share any more about that, since we do have a few minutes left.
Oh, sure. Yeah. So a lot of these discussions, were kind of advices from Dr. Hansen or Dr. Huang, but, if there is a way that, so I see kind of this opportunity as twofold. One is obviously for the trial itself, whether or not rapamycin has an impact on periodontal disease. The other aspect is maybe providing our senior care population who don't have routine care or dental oral health care, is to provide that opportunity for them. So, obviously the dental school here, and I know the senior care clinic is in Harborview. And so really it's twofold. But the way I see it is, obviously if individuals are, and once we get all the processing done, individuals are interested in taking part of the trial, obviously recruitment is something that we could collaborate with. But also if there are older adults that are in need of care or seeking care, we could hopefully also, create a little bridge between the two departments and maybe see some of those patients actually in the UW system, because a lot of times it's easier, especially if they don't have a dental home, to kind of help with that. And that's the other thing too, especially with periodontal disease, is that, you know, it goes underdiagnosed, as well as, you know, it's a lot of times patients don't know that they have it. And so they just end up, you know, losing teeth, and it could be prevented, or at least, you know, stopped. So I see collaboration is two-way. One is for the actual trial and seeing some, and if there's any systemic interest in terms of the trial, you know, please contact me in terms of looking at other assays that you want to maybe potentially look at. But also the second part is for our patients as well, in just around Seattle and Washington. So.
Most of our patients in senior care, I find anyway, fall into two buckets, which is they already have established care, which has been longstanding and is doing the best they can, or they're so advanced that all you all can do is full mouth extraction and then hope that we can find some pathway to dentures.
Right.
So, but yeah, send us your contact information for that. I have another question. Well, are you looking at the microvasculature of the gums at all in your?
No.
Anyway, well, you can contact me offline. That's our expertise. So
no, that's a great question, mate. Because this morning, I was writing some grants, and one thing we thought about, and this might be a line of your work, but a lot of the circulatory system actually goes also through senescence. So you could imagine some interesting things there. And I just laughed because I was trying to think of a way to do it, and you kind of asked. So I'll contact you later.
Yeah.
Okay.
Great. Yeah, I have so many patients in senior care clinic who have challenges in accessing oral health care, and it's so important, of course. So it's very exciting to hear that, and I think more bridges between oral health and geriatrics and primary care is just so important, and way too siloed. So this is exciting.
Absolutely. Yeah.
Great. Well, we're almost at 9 o'clock. I really appreciate the time you took to talk to us today. It was a fantastic talk, and you answered so many questions, and it's much appreciated. And thanks to everyone for your great questions. I imagine if people have other questions or are interested in collaborating, that they could reach out to you for that. So
I'll leave my email with Diane, so just contact me. Yeah.
Wonderful. Well, thanks so much, and take care, everyone, and congratulations again, Dr. Ahn. I hope you get some rest soon.
Thank you.